Next up, we have oral presentation 18, Jackson County, Georgia, Mission 12 Flight Experiment.
Our project is the hatching rates of Artemia selena in microgravity. I'm Audrey.
I'm Anna.
I'm Audrey Waters.
I'm Anna Holly.
And I'm Ella Cobb.
The purpose of our project is to identify a protein source that can reproduce in space. This protein can be digested by astronauts to prevent muscle degeneration.
Muscle degeneration is an enormous challenge for our astronauts today. Muscle degeneration is caused by not using your muscles for long periods of time. In microgravity, astronauts rarely have to use their muscles because the lack of gravity does not require them to. Scientists test muscle degeneration on the calf muscle, since this is where the majority of the degeneration happens. 70% of Artemia selena are protein. Protein contains amino acids similar to those found in human muscles. If astronauts eat the right supplements of Artemia selena each day, we believe their muscles will be replenished and stabilized.
Our hypothesis is that if Artemia selena are sent to microgravity, then the hatching rate of the cyst will be equivalent to the hatching rate in gravity. If our hypothesis is correct, then the astronauts will be able to grow their own protein source while in space.
For our experiment, we will be using 20 Artemia selena cysts, 6 milliliters of saline water, and 2 milliliters of 70% isopropyl. We will be using an FME3 tube for this experiment. For the crew interaction, Volume 1 will contain 20 dormant Artemia selena cysts until U5, when clamp A will be released and shaken gently for 35 seconds. This will mix the cysts with the 6 milliliters of saline water solution in Volume 2. This action should stimulate the hatching of the cysts. 2 milliliters of 70% isopropyl in Volume 3 will be unclamped on U2 and will be shaken vigorously for 10 seconds. And the fixative 70% isopropyl will be introduced to stop all growth of cysts in Artemia selena. The experiment will be conducted at the same time on Earth, and an analysis of the cysts will be completed to determine if Artemia selena in microgravity hatches at the same rate on Earth. If the Artemia selena do hatch in microgravity at the same rate, one day NASA may consider sending the cysts to space to help meet the protein needs of astronauts.
Once our experiment returns to Earth, we will use a microscope on the hatched or not hatched cyst from the mini-lab. We will compare those hatched on Earth to those hatched in microgravity. Once both experiments in microgravity and gravity are analyzed, a determination by comparison of the two experiments will be made.
We'd like to thank the Georgia Space Grant Consortium, the Georgia Institute of Technology, Ms. Hanley and Ms. Wright, the principals of Gum Springs Elementary School, Ms. Amy Tennell, the gifted teacher of West Jackson Elementary School, Julia Hoskins, the NanoRack staffer, Ms. Mary Belk, the executive director of the Crustacean Society, Mr. Robert Mangino, our SSEP editing assistants, Dr. and Mrs. Riddleberger, our research assistants, and so much more. And a special thanks to our teachers and families. Thank you.
Hello, my name is Valkyrie. I'm from New Jersey. I would like to know what made you guys think to use Artemia as a protein supplement. I know a lot of work has been done with Artemia in the past, so what made you think to use it in this way specifically?
Well, while researching, we found that, as you know, they have protein, a lot of protein. Specifically, 70% of their body is protein. And in that protein they have is amino acids, very similar to human muscles. So we thought that if the right supplement of Artemia is ingested by the astronauts each day, it will replenish the muscle loss that has happened that day.
All right, thank you.
Welcome.
Hi, I'm Ryan from Stamford, Connecticut. I was wondering if you know how many cysts need to be grown in order to provide sufficient protein to astronauts if the experiment is viable.
We don't know. We don't know the exact amount. We can research extra about that for further research.
Also, it would determine on their body weight what supplement it would be. So,
Thank you.
Hi, I'm Caitlin from Atlanta, Georgia. You guys did really good. So I was wondering, did you guys send the proteins to space yet?
no, they go up tomorrow.
Oh, wow. Congratulations. That's so awesome. And then how long are they going to be up before they come back down and you can continue your experiment?
5 days?
Oh, wow. All right. Well,
Or
thank you.
4 days.
4 days. Wow. Incredible.
Oh, 4 weeks.
4 weeks. Okay.
Sorry, I didn't know.
Well, very cool. Very cool. Thank you.
Hi, I'm Tim Livengood with NCSE. I have a kind of a basic question. So, Artemia salina, aka brine shrimp, aka sea monkeys. If we're suggesting that these are a food source, are they tasty?
We were actually looking forward to tasting them very soon, but so far we don't know.
Because I would think as an organism that grows in a hypersaline environment, you would need to do a fair amount of rinsing to get the salt out after you process the little things, even if you make them into a delicious paste.
Yes.
So, I'd be interested in that. Thank you.
You're welcome.
All right, thank you, ladies.